Good morning, everyone. My name is Steve Smyth. I'm a physicist in the School of Medicine at the University of Leeds. I'm also a member of the leadership team for part of the National Institute for Health Research, and it's my pleasure to welcome you to the Physics of Life and Rosetrees Trust Interdisciplinary Workshop on Neurodegenerative Diseases of the Brain. The aim of today is to encourage new ways of thinking about some of the grand challenges, if you like, in neurodegenerative diseases. And we hope that what will result from today's meeting are new and stronger collaborations across the disciplines, particularly between clinicians and physical and biological scientists. Now, this is particularly timely because some of you will have been aware of a strategic priority funded call to build collaborations at the life sciences interface. And we anticipate the second funding round to be announced within the next few weeks. So it would be terrific if, as a result of this workshop, some grant applications were to be submitted to that interdisciplinary call. I'm particularly delighted to acknowledge the enthusiastic engagement from the major funders of dementia research in the UK. And indeed to recognise that the Physics of Life Network and the nested Physics of Medicine Network, about which I'll make a few comments in a minute, are funded by UKRI and the Rosetrees Trust, respectively. The Physics of Life Network is now in its third cycle. The intention of the Physics of Life community is to drive collaborations between the physical and biological sciences, to consider some of the grand challenges at the life sciences interface, and to do so in a new way. So we consider a measure of our success the extent to which we're drawing in people from disciplines, perhaps to one slightly distant from the life sciences interface, and to draw them into grant-funded activity. The Physics of Life Network has funding to support workshops, sandpits and summer schools. I'm particularly pleased to note that we plan a summer school on a range of topics in biomedicine to be held at the University of Durham next year. And it would be super to see some of you and some of your students and some of the students on this call attending that summer school. And I would particularly encourage all attendees to consider applying to the Physics of Life Network for funding to run interdisciplinary workshops across the life sciences. We have funding to support that and follow on grant writing sandpits. We've also got some funds to pump prime research ideas, which are very much in the spirit of interdisciplinary working. Now, I mentioned that UKRI had funded the Physics of Life Network for the third time, but there's a significant and new element in Physics of Life, which is we call the Physics of Medicine Network. I'm delighted to acknowledge the support from the Rosetrees Trust for this element of Physics of Life. And the Physics of Medicine Network is embedded within this broader Physics of Life network. The core of the program, as you can see, are 6 deeply interdisciplinary workshops tackling a range of major clinical research questions. And we've had the first of these workshops back in November, organised by the University of York, on the physics of brains. For those of you who missed that, there's a record on YouTube, and we have written up, courtesy of an excellent science writer, we've written up the workshop by way of a sort of op-ed piece that we will be submitting to an interdisciplinary journal fairly shortly, and we plan to do the same for today's event. You can obviously find out more by looking at our website, and there's some material there that hopefully you will find of interest. Karis Baker, an academic at Durham University, is our network coordinator, and is very keen to receive comments and thoughts, and indeed suggestions for workshops. Our contact details are there. And of course, you can follow us on Twitter. we have a membership, but it's not a particularly complex or difficult arrangement to become a member. We would encourage everybody attending today to join up. It's simple to sign up, and you get access to a list of network events and opportunities of interest outside the network community. I think the important point here to note is that hopefully through the website, we can put you in touch with disciplines and folks from other disciplines that perhaps might be out with your normal community or contact list of contacts. This is the team running the network. I'm indebted to Martin Kahn, who's Professor of Biological Sciences at Durham, and Tom McLeish, who's the network chair. Tom's Professor of Natural Philosophy based at York. So on to today. I'm really thrilled that we've managed to secure some really outstanding speakers, leaders in the field. Sadly, I have to report a change to the programme. Giles Hardingham had a sad change of family circumstances yesterday and has had to withdraw from the meeting. But we've just managed to tweak the programme a little bit, and I'm indebted to Paul Matthews, who's going to talk for a little longer than originally planned on interdisciplinary approaches to neurodegenerative diseases. If we look at the next panel, you'll see that we're ranging across the field, and what will become apparent, hopefully, is the mix of disciplines that we've got. The speakers. I'm also delighted that we've got a funders panel who will be thinking about ways of supporting these novel and deeply interdisciplinary approaches. So we're grateful to our funders. And then moving on. In terms of the choreography or the etiquette of the meeting, we would strongly encourage you to use the Q&A function, the Q&A tab, because the panel discussions that you see in the program will be a particularly important element of this meeting. It's really there that we get particular stirring, if you like, of the disciplines to consider some of the problems. So please do participate. And then I think the final slide is about what happens after the meeting. Of course, we're all, well, certainly I'm missing the valuable face-to-face informal chats, and we've recreated this, we hope, in a virtual sense using a thing called the Wonder platform. And we would encourage you to stick with us after the meeting. You'll be sent a link, and your avatar will wander the virtual conference hall and be able to link up with the speakers and indeed other attendees of the workshop to explore ideas. So, and Karis has put some helpful links and notes about that in the chat. So that's enough from me. I hope you've got the sense of the aim of the meeting. We look forward to a stimulating day. And I'm really indebted to our Paul Matthews, who's the chair of the next session, and will also be speaking to us. Paul is a very distinguished clinical neuroscientist. He's head of the Department of Brain Sciences at Imperial. He led the very impressive UK Biobank brain imaging work, and he's an associate director of the UK Dementia Research Institute, which, as you probably know, is one of the major globally leading research platforms in dementia. So, Paul, thank you.
Thanks, Steve. And it's I who am indebted to you and your colleagues for helping to organize this fantastic meeting. Just give me a moment to get my slides together. So let me start off this section on interdisciplinary approaches with just a few obvious observations. And they're about observation. Observation is at the heart of science. And it's through novel vantages that we reveal new aspects of a problem. So that's really the logic behind interdisciplinary approaches, because they allow tools, analytical strategies, and modes of thought from different areas to converge. And these, in fact, have underpinned the successive advances that we've seen in neurobiology and disease, by and large, over the last 3 decades. Let me discuss some of the challenges first, and then I'll give you some illustrations of some exciting ways that interdisciplinary approaches have had an impact on our understanding of neurodegeneration, or will, and we'll hear many more through the day. But let me start with a really fundamental point that clinicians learn early in their training, and that's the difference between a syndrome, which is a collection of symptoms and signs shown by people who present with dis-ease. And a syndrome is distinct from what a scientist calls a disease, because a syndrome is non-specific. Many diseases can present with the same syndrome. For example, muscle weakness. Dementia is exactly the same. There are many types of disease that contribute together to the syndrome of dementia in late life. Here you see some of the major forms. The most common is Alzheimer's disease, vascular disease, and then you'll hear more about frontotemporal dementia from James, and there are a variety of others, including those related directly to environmental exposures, such as those related to alcohol after traumatic brain injury, and then rarer forms of primary neurodegenerative diseases, such as dementia with Lewy bodies and Huntington's. However, even Alzheimer's disease is a broad collection of specific disease entities, each with related but distinct causes. And these are reflected in the different forms that present clinically. About 13% of Alzheimer's disease is earlier in onset. Those are the forms most commonly that are genetically determined, familial or sporadic. The late onset disease is where the bulk of the clinical problem in the population lies. About 90% of people who have Alzheimer's disease. And this too is composed of a few people with familial forms that are genetically determined in a Mendelian or simple genetic fashion, and a very large group that have a complex genetic etiology. The interaction between literally hundreds, maybe thousands of different genetic variants with environmental exposures. So that's a real challenge for interdisciplinary science, how we can unpick that heterogeneity. But the problem is even more complex. And this is one of the greatest unmet challenges, because we are getting better at understanding what the different types of diseases are. But what we are still not very good at is studying the disease before its clinical stage. This is really important, because we've learned that in the case of Alzheimer's disease, which is what I'll focus on, the biochemical changes that begin to occur in cells of the brain start 20 or more years before the clinical expression of the disease, as changes in thinking and memory that affect daily life are appreciated by patient or family. Understanding when this biochemical stage begins, what its causes are, and distinguishing, when we look at the disease here, what the causal changes are from the cascade of secondary consequences of those causes, is a real challenge. We need to find ways of identifying people in this phase, characterizing the changes in their brains at a system and cell level, and then monitoring its progression here to understand this cascade of events, so that we can distinguish these clearly, intervene and stop the disease before it develops clinically. Now, I can frame it in that broad way but what it highlights is that we really have major conceptual challenges in neurodegeneration research for which we urgently need solutions, and we need to reach across to those in other fields to help us. Let me highlight what I think are the 6 major conceptual challenges. The first is not just how can we discover the stages, how can we discover the different forms of dementia, but how can we discover them early so we can distinguish mechanistically distinct diseases from their initial stages. The second is, as these, having discovered that there may be different processes, we fundamentally still don't really understand how brain circuits fail. We're beginning to get some insights, but we really need to piece together which cells are involved and when they're involved. What are the first cells to be affected in Alzheimer's disease? What follows and why? And this really gets down to the key point. What are those mechanisms that drive dysfunction in the initial stages, in the initial cells, and what are the mechanisms by which the cascade is initiated? And when? Does it start 20 years before? Does it start when someone is born? Could neurodegenerative disease of late life be a process through the life cycle? These are really important because we need to ask ourselves how we can stop it and when we can intervene to have the greatest impact. Underlying this challenge is a real technological and further conceptual challenges, and that is that these questions cross multiple scale spaces. We need to think about the scales of dynamics in the organism. I've rather just highlighted this in referring to moving from cell-cell interactions in the early stages of the disease back to actually understanding life course. We need to be able to see disease progression over periods of time much longer than most of our scientific careers. We need to understand the way cells talk to each other in the body, not just in the brain, and over multiple time frames using hormonal signaling and endocrine factors, trying to understand why, for example, the risk of neurodegenerative disease may change between men and women. We need to understand the chemical signals that pass between cells, between neurons and glia, between glial cells. We need to understand at a finer resolution how within cells or between cells competing pathways are controlled, how metabolic regulation is functional and dysfunctional in the context of the early disease. And fundamentally, certainly in Alzheimer's and many of the neurodegenerative diseases, we come down to the problem of abnormal protein folding and how this is mediated, how competing folding pathways shift their balance in the initiation of disease, and how they can be modulated to limit the spread of disease. We need to in doing this, we need to look across spatial scales. I'm going to ignore the effects of environment, which is a very big spatial scale. Let's just focus on the body. We need to understand not just how brain cells are interacting, but how the body is interacting with the brain, how the immune system of the body, for example, how the metabolic system of the body, how the gut influence the brain. We need to understand how brain regions communicate. One of the most striking things about Alzheimer's disease is that there's a regular sequence overall in the anatomical progression of the disease from one region to the next, and so on. Why? How? There are elements of circuit dysfunction that occur that mediate the cognitive impairments that we're beginning to get a broad description of, but we don't really understand. Why do circuits begin to go wrong? What circuits are affected? And of course, the circuits are driven by cell interactions, cell-cell interactions, neurons to neurons, of course, glia to neurons, glia to neurons, vascular compartment to glia to neurons. We hear, and then within cells, key to much of what goes on in Alzheimer's, it is becoming clear from the genetics, lies in interactions between processes involved both in lipid homeostasis and in protein homeostasis involving endosomes, mitochondria, and in turn their interactions with synapses. Well, interdisciplinary approaches clearly can help with many of these problems. And let me just highlight very, very quickly a few of those areas in which I've seen evidence for some real impact. Already, in fact, the low-hanging fruit as a consequence of the rapid growth in computational power over the last couple of decades has meant that large data sets have been able to be aggregated and modeling using these large data sets, linking the data sets, developing predictive approaches based on one set of data informing another. And these are beginning to make their way into the clinic. Let me give you a description of how this is beginning to be realized. Small, rather focused data derived from deeply phenotyped patients studied in cohorts have been used to develop models, models of disease treatment response. These can be used to develop hypotheses which are expressed through analytical models developed either statistically or through AI techniques or direct mechanistic causal modeling. Now, these tools, these models, with increased computational power can be brought to bear on big heterogeneous data sets, such as one finds from routinely available clinical data aggregated through electronic health records, in which surrogates of specific focused observations done under research conditions can be identified, and the models applied in a looser way using much broader data in which smaller signals can be defined to test hypotheses on bigger populations for their generalizability. With these tests, with the models embedded in tools for large-scale computation, these in turn can be used to drive clinical decision support systems in which clinical experience, rather than being anecdotally managed by individual doctors as they go about their daily business, can be embedded in quantitatively defined experience over a hospital system, multiple hospital systems, global experience with the disease to give likelihoods of clinical outcomes with specific decisions. It is already beginning to happen. It's already being used, particularly in some areas, to support pharmacovigilance studies after marketing. Well, scale space, sometimes there are some really brilliant examples, and we'll hear a few of those later today, of ways in which analytical strategies alone have been used to cross scale space. And I'll give you just one quite elegant example from Steve Smith in Oxford and his colleagues in using analytical strategies to cross between the scale of global organ senescence measures or global organ brain aging, in this case for the brain, down to molecular hypotheses of specific genetic determinants of these processes. What Steve and his colleagues did is looked over the UK Biobank data set and over very large numbers of subjects. They were able to describe age-related changes using multiple brain imaging measures. From these, they were able to describe a global brain imaging change, and more importantly, how individuals deviated, the extent of their deviation from these global age-dependent changes. And these deviations embed the concept of either senescence at the same rate as the mean, senescence at a lower rate than the mean, that is to say slower aging, or senescence at a higher rate in the mean. Now, what they did, which was particularly elegant, is they looked at this from the standpoint of the analytically defined multiple multivariate components of this aging rate. And they found that populations, it's not just that populations show variable aging, but that populations show variable aging across feature clusters. They then were able to use these feature clusters to show how they could markedly improve the power of genome-wide association analyses for identification of possible molecular determinants of these changes in senescence properties of the brain. At the bottom is described what you see are chromosomes arrayed along the abscissa, significance of feature association along the ordinate, and with the global feature of brain aging, there really is very little that crosses the Bonferroni significance line. But when they look at very specific brain aging cluster trays, they see a very different feature. And this really highlights how specificity of phenotype description, which is something that interdisciplinary science has been very powerful at driving, hugely enhances the potential power of explanation in disease biology. I won't go into any of the individual findings. The second really exciting example came out in Nature last month from the Allen Brain Institute laboratory, which has led so much in interdisciplinary exploration of the brain. And this is something that I think holds great promise for our understanding of disease biology as it moves forward. And here they combined a theoretical understanding of the potential hierarchical arrangement of visual processing in the brain with a specific dissection of anatomical hierarchy in quantitative terms based on cellular connectomics, in this case, synaptic densities, mapping out the feed forward and feedback pathways across different regions in the mouse visual cortex. You see the lateral geniculate nucleus, various cortical regions arrayed above it. The connectomics, anatomically described, was able to be summarized as a hierarchy score from which expressed feed forward and feedback circuits. What they then were able to do is link these hierarchical scores with measures of function, and they showed that anatomical hierarchical measures were strongly correlated with the functional measures. And the power of this is the precision of the analysis allows its specific dissection in the case of changes in any of these functionally important elements to understand causal mechanisms at an anatomical level. Another example where interdisciplinary science is exploding right now in helping us understand relationships between cells and molecular events, moving down one level from systems, has been in the revolution in single cell omics techniques that we've seen over the last 5 years or so as a direct consequence of improvements in bead-based chemistries, microfluidics, and then the increases in efficiency of sequencing to the point now where it is a routine process to sequence literally tens of thousands of cells or nuclei from tissues in a day, from which one can develop detailed descriptions analytically in the cellular populations being interrogated from their transcriptomic signatures, and from these transcriptomic signatures, taking a deep dive, one can look at gene co-expression maps. From these gene co-expression maps shown here in the background, one can highlight genes of interest. In this case, for neurodegeneration, the genes that have been the major genes identified by Alzheimer's disease genetic association studies, and potentially therefore causal in the disease, to understand their co-expression networks in a given cell type, from which one can look at the cell-specific patterns of expression and infer cell-specific functional roles for these genes that are putatively involved in the early disease. It's a beautiful example being exploited by many groups very rapidly. We can begin to relate these cellular events and walk up the complexity tree further by using methods where we can highly multiplex targeted protein expression. Here you see a limited number of these. Just for illustrative purposes, this is imaging mass cytometry, which is a scanning mass spectrometry technique that detects tags of heavy metal ions on antibodies, allowing hugely multiplexed imaging of targeted proteins across tissues to look at spatial relations between different specific types of cell markers. One can take that one level down to to relate directly to the kinds of maps I showed you on the cellular transcriptomics a moment ago, using spatial transcriptomics techniques, which is an extension of bead array technology, looking at an array of beads across a glass plate, and on each bead performing a single cell transcriptomics approach, from which one can highlight the distribution of genes of interest in their spatial relations, which can in turn be related to protein expression in highly multiplexed fashion to understand system, cell, gene, protein relations. Much of what we want to know about the relation between genes, cellular function, and cell interactions lies at the subcellular level. Mass spectrometry, again, in one of the most elegant examples of interdisciplinary science I've seen in recent years, can be used to probe this in really exciting ways. This is from Ian Gilmore's group at the National Physical Laboratory, using 3D orbisims, a high-resolution mass spectrometry mapping technique, in which he's been able to show how he can map the distribution of specific fragments in mass spectrometry, which can in turn, of course, be related to specific molecular species across at the level of tissue resolution, but most importantly, take a deep dive with this method, look across specific cells, and most exciting, move the resolution down to just to several microns, allowing subcellular interrogation of distribution. This is a way that we can actually begin to go from the level of broad anatomical systems to cells to interrogate what's happening at the level of subcellular organelles. And of course, signaling between cells, the challenge is everything I've talked about recently is static. We need to find ways of developing dynamic approaches that can work in real time in vivo. It's a real challenge for humans at this point still, but some incredibly exciting approaches have been proposed in animal systems, and these have been fueled by hugely interdisciplinary work in which, for example, this lovely example from Caltech shows how AI-driven synthetic approaches, which I won't describe, but they fundamentally rely on understanding small molecule protein interactions using AI methods to design proteins to fit, small molecules to fit protein pockets in specific ways, were used to develop a sensor molecule that could specifically respond, here's the polypeptide sensor, to the presence of serotonin, provide a strong fluorescent signal from this, which could be, and with the expression of the polypeptide in the animal using transgenic methods, they were able to dynamically monitor spatially using optogenetic methods the release of serotonin over time. Here you see serotonin spiking in a regular way. It's blocked with escitalopram, and really, really exciting. Now, the final thing I just want to touch on before I close is one of the most positive things in a very difficult year, and that has been how another aspect derived from interdisciplinary science, which we take for granted now as communication, has utterly changed the game. With the onset of the COVID crisis, the scientific community has mobilized as never before have I seen it in my lifetime. And we've seen how scientists of all disciplines have joined in different ways to provide insights into SARS-CoV-2 and its and its consequences. And one of the really important frontiers for those of us interested in neurodegeneration is understanding what the mechanisms are through which the virus may be causing neurological consequences, and what the long-term effects of this may be for neurodegenerative disease. Here are some fascinating results from organoid models showing infection of the organoids with the SARS-2, which you see in green, and you don't see in the untreated organoids. Moreover, one can see expression of tau induced in the neurons in these SARS-treated organoids, and it has provided the clearest evidence that we have for a model in which viral infection leads to expression of tau and neurodegeneration when it's found. So I'm going to close here by just pointing out a really nice paper by Eugene Duff from Oxford recently in Nature Neuroscience, which discusses in a very useful way the challenge of how we can, if we do start to move across scale spaces, how we begin to develop representations that are useful for us to make inferences from, how we can develop these, how we can understand variability, express generalizability, and ultimately lead to something that is meaningfully interpretable. So let me just close by saying again how important interdisciplinary science is to neurodegenerative disease, how important it will be, and leave with just an appeal to everyone listening to consider how they can help with this urgent agenda. There are 850,000 people in the UK with dementia now. There are going to be 1.6 million people with dementia in 2040. And there's one new case diagnosed every 3 minutes. It's time that we all got together for this and related clinical problems to make a difference. Thank you.
Paul, thank you so much for a really outstanding discussion, presentation, and it will lead to an outstanding discussion, I'm clear on that. There are a number of questions have been raised in the chat, and we will come back to those when we come together as a speakers panel in a few minutes. But thank you again for a really terrific presentation, which is clearly demonstrated, so clearly demonstrated the value of interdisciplinary collaborations. Can I just invite you then to also chair? We're getting our money's worth from you, Paul, this morning, but I'm very grateful that you've also agreed to chair this next session. And as I say, there are a number of specific questions being raised in the chat to which we will return in the panel discussion. So thank you, and back to you.
Thanks, Steve. No, it's a great pleasure to move on to the next three speakers and the first is James Rowe from the University of Cambridge, who's well known particularly for his studies of frontotemporal dementias and related syndromes. James is going to speak to us today about variance, and veracity. That's a tongue twister. Clinical challenges for dementia research. James.
Thank you, Paul. Thank you for a kind words and introduction. I'm going to pick up on many of the themes you introduced. First, for the records, I've no disclosures that are relevant to the material that follows, but I'll start where you left off. This is a pre-COVID picture, we remember these days, with the scale of the challenge of dementia. 1 in 3 of us are heading towards dementia as things stand. But it's a very individual experience, genetically and biologically. We'll all be different, and it'll have a different impact on who we are as people, our personal autobiography, our social communication, language. It's a very individual experience. And this raises a number of challenges, which I've listed here, or started 2 of them here. One is, how can the molecular and genetic discovery science that is moving apace so quickly, as Paul highlighted, help people who are affected by dementia? Related to this question, how can precision medicines and medicine trying to target that individual nature of the dementia experience and dementia biology, how can this be generalized? The first thing I'm going to really set out today is that we need to seek precision, not just at the molecular or genetic level, but also at the phenotypic level. And we also need to have clear and precise models of what I call intermediate phenotype, linking causes to symptoms, providing targets and outcomes and mechanisms to support clinical trials. So just to illustrate the problem, we take the commonest dementia of Alzheimer's disease. Familiar in its amnestic, its memory form for many cultural icons, presidents, prime ministers, and so on, the dominant form. But you may also have heard there are variants which give visual loss, made most famous by Terry Pratchett, also which have a motor control syndrome caused by Alzheimer's disease, really brought to light in this excellent book. But there are other variants that you probably haven't heard of that are much less well known. So we have the problem that patients who have the same diagnosis, the same label, the same disease biology, may actually have no symptoms in common. So people often say, well, what we need is to have to rely on the molecular and genetic precision, and this will be the answer. And I'm going to challenge that assumption. Taking, for example, the problem of genetic pleiotropy. So illustrated here with mutations of a gene called C9orf72, has a very precise molecular signature with dipeptides, repeats. At post-mortem, there's a very particular, very characteristic TTP43 type pathology. So a lot of precision at the molecular and genetic and pathological cellular level. The problem is it causes a myriad syndromes, different disease profiles. I've listed some of the commoner presentations here. So there really isn't a direct link from that genetic cause with its singular pathology and molecular characteristics to the nature of the illness that the people actually have. The problem is reversed. If we take, let's say, some of the commoner presentations, we try and link these syndromes back to that molecular pathology of tau and amyloid and alpha-synuclein and so on that we see in histopathology so clearly. We find that each of these syndromes may have many different molecular causes. So there's no easy mapping from gene to molecule to disease syndrome that the patients are affected by. So we need a different strategy, and I think I'm going to argue that we can aim for a precision at the level of the clinical phenotype, asking really what the patient actually has, not what they're called, not the diagnosis, what is actually going on with a given patient. This calls for an approach that looks across different diagnoses. I call it transdiagnostic methods, and it embraces the variance. It says that the variability from person to person is actually informative and important to understand. I'm going to illustrate this with a study called the PIPIN study. It's a pick disease and progressive supranuclear palsy prevalence and incidence study. It's been running for a number of years, trying to trace all people with syndromes, the different variations of phenotypes associated with a group of pathologies and genetics known as frontotemporal lobar degeneration. It's an epidemiologically based study in the east of England, trying to trace every participant. And we take detailed phenotypic mappings, so detailed analysis of what symptoms and signs individuals actually have. So this is based on some multidimensional scaling, looking at the relationships between the presence of one symptom and the presence of another across all these participants, 365 people. Now, I've color-coded them post hoc according to those symptoms that underlie the classical diagnostic labels. But if you look beyond that color coding, you actually see there's a fairly smooth distribution of symptoms and signs. And although we might see this cluster here that I've made easy to see by blue may look like typical progressive supranuclear palsy, or the cluster of features in red are typical frontotemporal dementia, there is no clear blue water between these symptoms. There isn't actually discrete representing discrete disorders. And patients may exist anywhere on this two-dimensional here. Let me just change my pointer for a moment, sorry. So you may have a patient who has this cluster who would span two different diagnostics. You might have a patient here with this cluster of symptoms, with, in a sense, meeting multiple diagnostic labels, discrete entities, each represented here by, to much more of this picture, a smooth and continuous high-dimensional space, represented here as a color space. And any given patient may exist anywhere on this high-dimensional map. The vertices represent the classical syndromes, but actually patients may exist anywhere in this space. So we need a transdiagnostic approach, and we can be precise in their phenotype and then start to ask how and why these phenotypes matter and how do they arise and what we can do to treat them. So just to highlight the importance of these phenotypes, if we take from the PIPIN study challenging behaviors of apathy and impulsivity, these are a defining feature of the behavioral variant frontotemporal dementia. And indeed, patients with this diagnostic label score very highly. But these same clinical features are not diagnostic features of these other diseases. I'm sorry about the name, just take these as 5 other different diagnostic groups. But we can see if the patients here in the blue and green who've got progressive supranuclear palsy or cortical basal syndrome, about half or 2/3 of them express these behaviors to the same level as patients with frontotemporal dementia. So this is a feature of disease that spans multiple diagnoses. Does it matter? Yes. This phenotype, the expression of these behaviors, regardless of your diagnostic label, has a huge impact on your survival. This is a survival curve of independent survival, time to nursing home essentially, or death. It's as stark, if not more so, if we look at simply time to death. So this phenotype is really important, and this really should be our target when we're thinking about understanding and treating disease, trying to improve quality of life and health impact, reducing the need for institutional care and prolonging quality and duration of life. So let's think about aiming for the target. Well, I'm going to say our target is really about the experience of illness, quality of life, and survival based around those behavioral and cognitive features. And I've highlighted there is not a direct link to those root causes, but we can try and build mechanistic causal relationships, building up the layers of cause and consequence to link those root causes to the outcome that we're interested in. But if we want to change and improve this final stage of quality of life and survival and health economics, we should really put a lot more attention at aiming our understanding of disease and treatments at this level that is close to the target, close to the goal, and not only focus on this more distal underlying remote causes. So keep this cascade in mind. I'm going to just put it up here and as a reminder. So we're going to say, how can we build precision at these intermediate layers? I'm going to illustrate this with 3 examples. First, understanding the variability of GABA deficits. GABA is the principal inhibitory transmitter in the brain. We're going to show how this can be affected across multiple syndromes and provides a candidate target for treatment in its own right. So GABA is reduced by frontotemporal dementia and progressive supranuclear palsy. And perhaps one of the initial links here to the physicists in the audience is that to demonstrate this needed to move from standard possible scanners at 1.5 or 3 Tesla MRI up to ultra high field 70 MRI for spectroscopic precision and increased sensitivity. With this ultra high field MRI, we can see a clear difference between patients, healthy patients, patients with either disease and healthy controls, showing that some people, not everybody, some people have reduced levels of GABA. The paper here with Alex Murley went on to show that this loss of GABA also correlates with the severity of the syndrome, particularly impulsivity, showing its clinical relevance. So GABA is reduced, and this accords with post-mortem biochemical assays in these patients. But what is the consequence of this on cognitive physiology? Well, to understand the consequence, I'm going to turn to magnetoencephalography to understand the physiology of dementia. And the background here is to understand that when cells are active, there are postsynaptic potentials and currents that run down the apical and principal dendrites of neurons. These generate a perpendicular magnetic field that traverses the skull directly and readily. And this can be detected with highly sensitive magnetometers and gradiometers, which require supercooling, so they need to be held here in liquid helium. But they can still be within a couple of centimeters of the skull and therefore detect these ultra-sensitive magnetic fields pertaining to active neurons. And this provides a very safe and well-tolerated environment to study the brain, and it gives very good agreement with direct recordings with intracranial EEG that we've conducted with similar Tarston paradigms. And it provides an ideal basis for experimental medicine to understand individual differences. So let's look at an example of this. So looked at a phenomenon known as beta desynchronization. It's a change in the beta oscillations in the frequency range here. These are critical for state transitions for movement and cognitive flexibility. And this beta desynchronization is impaired by both frontotemporal dementia and PSP, and it's impaired over the frontal parts of the brain. So I'm showing here in yellow the areas of the brain where there's a reduction by disease in bivalent frontotemporal dementia and PSP. Now I'm going to make a link between that and the changes in GABA and say if we were to prevent the breakdown or the reuptake of GABA, in other words, to extend the GABAergic transmission efficacy using a drug called tiagabine, it's a GABA reuptake inhibitor. What we find is illustrated in red here that in much of this prefrontal cortex, we restore the beta desynchronization. So we have a potential reversal of the pharmacological deficit that normalizes the physiology underlying a wide range of cognitive processes. So this method, it may tell us that there is a potential benefit, but it doesn't tell us how that tiagabine is working or for whom it might be working. And for that, we need a different approach. We need to switch to a different set of methods using detailed models of cognitive physiology of brain function. So to illustrate that here, I'm going to draw on the work and collaborations with Carl Fristner, who will be talking later today, and Ros Moran. And so we need to reformulate the brain, not just as a simple descriptive level, but with a model to say that each of these levels in the frontotemporal cortex can be conceived as a chain, a network of areas And within each network, we have a relatively stereotyped format of superficial and deep pyramidal neurons, spiny stellate cells, and inhibitory populations in a tightly regulated, what I might call a canonical microcircuit at each of these areas of the cortex. And this becomes a tractable problem if each of these cellular populations can be represented by coupled differential equations relating to the time constants of the principal neurotransmitter based synapses and membrane conductances. So this becomes a computational tractable problem to represent neuronal dynamics. And let's see how this works in practice to understand whether, and in whom, and how a drug like tiagabine is working. So we're representing our brain with a network, if you like, a chain series of these canonical microcircuits symbolized here. This is representing the right prefrontal cortex, an area where we have shown in these same subjects that they are deficient in GABA using 7 Tesla ultra-high field spectroscopy. So let's take these patients, and for each of them in this frontal region, we can build a biologically informed computational model of that local cortical function, showing how superficial and deep pyramidal neurons are regulated by inhibitory cell populations and spiny stellate cells receiving inputs from lower hierarchical levels. We can ask whether this model is able to, is valid, the veracity coming in here. And there's several steps we might take to do this. One is to ask, can this model reproduce those magnetoencephalographic observations that we saw measured with MEG and EEG? The answer is yes. So for 2 regions in the brain, under blue and red are 2 different drug conditions. We see the predicted and the observed time series are an excellent match. In other words, the model is capable of reproducing, of simulating the brain function during the task at hand. We can also show, if we take some of the connections, so this frontal to temporal connectivity here within the model, the strength of that connection correlates with clinical severity. This is a frontal cognitive battery. So again, it gives some veracity, some validity, clinical relevance of these models. And the third step is to then say, what about tiagabine? So if we look at the effect of this GABAergic manipulative drug on, for example, the synaptic scaling, the efficacy of inhibition, we see that tiagabine, as we would expect, increases the efficacy of local inhibition by inhibitory neurons on pyramidal cells. There's also one at a group level, but we're interested in who does this work and how. So we can then look at the performance of this model. I'm going to focus really first on this deep inhibition of the pyramidal neurons as an example of the level of cellular and synaptic specificity we can achieve with these models. So this inhibitory connection, which is restored at a group level by the tiagabine, but it doesn't have that effect equally in all participants. What we see is that the response to tiagabine, this GABAergic drug, is a function here in blue of that individual patient's GABA concentration as measured by the 7T spectroscopy. So we can start to see a state-trait interaction, if you like, that the trait of your intrinsic GABAergic deficiency caused by the dementia predicts the state or the acute effect of a restorative drug treatment. And just to highlight the specificity, which I find personally extremely exciting, is that the effect of a GABAergic, pharmacologically specific drug to restore cortical function depends on those GABAergic interneurons and as a function of individual GABA levels. So this is beginning to give pharmacological and phenotypic specificity in an experimental medicine context. Of course, the GABAergic drugs and GABAergic interactions don't take place in isolation. There's a context here that those oscillatory dynamics we spoke about, those beta oscillations, depend on very tight coupling of inhibitory and excitatory neuronal populations, particularly mediating NMDA to GABA interactions. So we could then look in these same patients using an alternative drug, memantine, which is an NMDA receptor antagonist. What about that in the context of these same patients? And here we show that the effect of memantine, which acts on the NMDA, is conditional on an individual participant's GABA concentration. So this is I'm showing here on the y-axis the change in the evoked response to a stimulus in patients between the placebo and the drug condition. That drug change in the evoked physiological response is a function of the GABA concentration, the alternative neurotransmitter. So trying to illustrate that we can try to bring precision at these intermediate levels, illustrated through the GABAergic drugs and NMDA effects, showing specifically this applies across multiple diagnostic groups, PSP and frontotemporal dementia in this case, providing some individualized predictions of treatment response. In the next few minutes, I just want to touch a bit on a second illustration of this using the neurogenergic system. So the neurogenergic system is extremely important for cognition and arousal, and all of the brain's noradrenaline comes from a small region in the brainstem called the locus coeruleus. It's a small region. It's rather like a short length of spaghetti, about 1.5 centimeters long, illustrated here. And this is very impaired in many diseases: Alzheimer's disease, PSP, progressive palsy, Parkinson's disease. Many common dementias lead to degeneration of the locus coeruleus. And we've shown over a number of years, for example, in this series of studies in Parkinson's disease, that you can use drugs act on the neurogenetic system, one called atomoxetine, for example, to restore behavioral control. But across all of these studies, there is a problem that the drugs seem to work in some individuals reliably, but not others. In fact, some people it made matters worse. So it's a highly individualized response to the drug. We want to understand how could we predict and target treatments to those people who are likely to respond favorably. For that, we return to the locus coeruleus and we say, well, what about individual differences in locus coeruleus function? So if we look at a post-mortem slice, this is a slice through the base of the brain at this level here, through the locus coeruleus, a horizontal slice. We can see at the back here, this little black dot. This is the locus coeruleus. It's tiny. It's about 1 millimeter or so across. It's dark without staining because of intrinsic neuromelanin. These are complex macromolecules containing iron, and it's visible to the naked eye. If we look under the microscope, each of these black dots is a single cell that's producing noradrenaline. Each cell is labeled, if you like, by its own neuromelanin. If we zoom in on these cells, we can see within them granules, these macromolecular granules. And the significance of these is these complex molecules, their protons interact with free water protons with a phenomenon called magnetization transfer. And this means that if we return to the ultra-high field 70 magnetic resonance imaging, we can have sequences that are sensitive to this interaction between the macromolecular bound protons and the free protons. And this gives us the ability to image the locus coeruleus. So showing here, this is on, again, a horizontal slice. This little red dot here is the same as what we see with the naked eye, this tiny but critical structure. And we can image and quantify the locus coeruleus, and we can show that in patients with Parkinson's disease or progressive supranuclear palsy, that the integrity, the contrast to noise in both of these patient groups is greatly reduced compared to controls. And that's important for individual differences in behavior. We can illustrate this through double-blind placebo-controlled drug studies here with that drug atomoxetine. I'm going to present the Parkinson's data first. The PSP work is still ongoing. And we're going to look at that impulsiveness, returning to this problem of the disease that was so important for outcome and health economic consequences. So for brevity, I'm just going to assert that the impulsiveness can be measured by this quantity called the stop signal reaction time. It's how much time you need to prevent a disinhibited action, how much advance warning. And we see that the response using this measure to the drug, the change from the drug between atomoxetine and placebo, varies a lot. There are a number of patients who are improved, and there are also those who are made worse. The critical thing here is there's this correlation, so we can predict and understand whether the drug will help or hinder an individual patient in terms of this locus coeruleus contrast to noise. So we're starting to be able to target treatments not by diagnosis, not even just by whether they have impulsivity or not, but that biological trait. And this will be built into a phase 2 clinical trial that should be starting imminently. We're very excited to take these previous experimental studies and move them into a clinical trial context in the next few weeks. So just to summarize where we've been, I've highlighted that we really should keep our goal on the target of improving quality of life and behavior, but we need to understand the behavior, not just in terms of diagnosis, but this multidimensional space where individuals with different diseases can be located. We also can use advances in technologies like MEG, magnetoencephalography, to understand the concepts in terms of cognitive physiology, the neural oscillations that underlie cognition. But we need a mechanism, we need a causal model, which can be provided by these generative models or computational models of local cortical function. These can predict and explain individual differences in the response to drug treatments, and we can refine that by exploiting some of these cellular and molecular characteristics measurable with MRI, and really take this directly into clinical trials. So just to my final slide, just to summarize, that we should celebrate and embrace the variance of diagnosis, the variance between patients, demand that there's veracity to the models and validity to our models, but ultimately be aiming for precision in that phenotype to guide treatments and building models of those intermediate phenotypes that link causes to the symptoms that we'd wish to treat. So I'd just like to thank you for listening and thank the many people in the group who made the work possible that I've been talking about today, and look forward to any questions in the Q&A session or offline later on. Thank you.
James, thank you. That was terrific. So moving to the next talk, I'm really pleased to introduce my former colleague and current colleague in Edinburgh, Craig Ritchie, who will give us a real clinical perspective on interdisciplinary approaches to developing new therapeutics. Craig, thank you.
Thanks very much, Paul. Can you hear me okay?
Yeah.
Okay. So, yeah, I just want to build a little bit on what I guess James and Paul were introducing already, but take it from the perspective primarily of some of the challenges we've had in developing new therapeutics, specifically for Alzheimer's disease. Now, I won't spend a lot of time, outlining the challenges that we've had over the last, say, 20 years or so in bringing new drugs to market. It stands, I think it's quite obvious to everybody in this community that we've spent billions of pounds or dollars investing in clinical trials of various agents. And as over the last 20 years, there haven't been any new drugs that have actually made it through to market to help patients with Alzheimer's disease. And I think there's one thing that this audience should give some consideration to, and that is that when we think about therapeutics for Alzheimer's disease, we do tend to group things into better, newer symptomatic agents that will help the clinical symptoms of a dementia syndrome or an early sort of mild cognitive impairment stage of disease. But what the primary focus has been, and probably quite rightly so, has been in disease-modifying therapies that will be used very early in the course of disease, that will be able to perturb disease mechanisms that will then, in effect, hopefully prevent dementia. Now, when Paul started his presentation, I was delighted to hear him say that observation is the most basic requirement of a scientific inquiry. And we hadn't talked about these talks together beforehand, but I've written, I think, accurate measurement or observation is the most basic requirement of any methodology. And through the course of the next 20 minutes or so, I'm going to sort of outline, in a sense, what some of those challenges are with measurement, which have probably had a deleterious effect on our ability to develop new therapies for Alzheimer's disease. And I think one of the things I'd like to talk about, and I think my previous speakers have spoken about this already, is this whole notion that the diseases that lead to dementia in later life, the syndromal diagnosis that Paul outlined, start at least in people's 40s or 50s, if not even earlier. And I've been fortunate enough to lead two very large cohort studies, EPAD, the European Prevention of Alzheimer's Dementia Programme, and the Prevent Dementia Programme, which is based in the UK and Ireland, which are really now beginning to bring some incredibly important data to articulate some of those issues or mechanisms of disease in the decades well before dementia develops. So, very simply put, if you're designing a clinical trial and we're testing an intervention, we need to make sure we're testing that intervention in the right population, and we have the right measures of success. Now, success can mean different things at different stages of the drug's development. It can mean different things to different populations, to regulators, to patients, to academics, etc. But one should think that these sort of 4 areas of measuring success by way of target engagement, perturbation of mechanism, impact on the disease process itself, and therein clinical benefit, should be as closely linked together as they possibly can be. Now, one of the big problems is that when we talk about modifying the course of disease very early on, we are doing so in effect in a preclinical population. Therefore, clinical benefit can't really be measured very effectively with the tools we have available to us. The challenge, and I'll expand on this later, is when we speak to regulatory authorities about modifying the course of disease in a preclinical population, they still want to see clinical benefit as being the primary outcome. And that, of course, creates some challenges. So I think we have to reframe or to rethink neurodegeneration and dementia. And this cartoon to illustrate it, I use quite frequently. It's a very simple cartoon, and it's just illustrating what was spoken about earlier, that it's highly likely that the diseases that lead to dementia in later life have their genesis at least in your 30s or 40s. In the earliest stages of these brain disease, we talk about there being a silent period before mild, moderate, and severe problems emerge. And it's only those severe problems which would satisfy the criteria for a dementia syndrome. The other issue here is that we talk about this silent period, and one could argue it's only silent in a sense because we're not listening properly. Because the tools we have in clinical practice, in clinical trials, and epidemiological studies aren't sensitive enough to pick up what's going on in the brain or in terms of neuropsychological function to be able to detect those changes. And I'll talk a little bit later from the PREVENT study about how we may be overcoming that. So Alzheimer's disease. Alzheimer described the pathology of Alzheimer's disease almost well over 100 years ago now in his patient, Auguste D. Now, he didn't describe it as a clinical syndrome. He described it, of course, a post-mortem on the presence of 2 key pathologies, and these are its original sketches. And that is the amyloid plaque and the neurofibrillary tangle. Now, the problem, of course, was in 1903, when this was originally described, is there was absolutely no mechanism by which these pathologies could be identified in life, in vivo, if you like. So what we had to do was to try and approximate a clinical phenotype to what was believed to be happening in the brains of individuals with Alzheimer's disease. But if we fast forward to 1990, when we were using imaging techniques, CSF biomarkers, we were now being able to measure Alzheimer's pathology, be it tau or amyloidosis itself, using imaging, CSF biomarkers. And of course, now we're at a very exciting stage, and I know Henrik will probably talk about this later, where we can use blood-based biomarkers to identify these pathologies in life. Now, I wasn't really around in the early 1990s, but one could have imagined, working academically, one could have imagined at that moment there could have been a kind of a eureka. We can now measure this disease. We can bring the brain back into the Alzheimer's disease, stroke, dementia, diagnosis. Unfortunately, there was probably too many legacy issues about now considering Alzheimer's disease as a memory disorder, as a cognitive disorder, to be able to pivot to then putting the brain front and center, if you like, of the clinical diagnosis. So this is where we have been really probably for the last, 30 or 40 years, that we've looked at these brain diseases through the peephole, if you like, of the older person with the dementia syndrome. And most of our cohort studies and our observations and our clinical trials to date have been in those populations. And that probably also reflects on the fact that our clinical practice is in those populations through memory clinics. Memory clinics tend to see people with a dementia syndrome or just in the few years before a dementia syndrome develops. So our learnings, if you like, about those brain diseases in life are limited by the populations we observe them in. What I would argue is, and over the last few years, is we've started to shift our gaze and being able to recruit people into cohort studies and therefore being able to look much more detail, phenotypically and biologically, at what's happening in the brains of these individuals and how they interact with risk factors and genetics, etc. And what, of course, we then do is pivot our thinking away from, if you like, looking at cognitive and behavioral and functional outcome measures, which are very difficult to measure in any regards in terms of accuracy, to be more reliant, I think quite rightly, on biomarkers of the disease in these earlier stages. I've mentioned a couple of the projects, the Prevent Dementia project, 700 people across UK and Ireland, aged between 40 and 59 at baseline. In the EPAD study, there's 2,100 people across Europe who've contributed to this, and all of those people in EPAD have CSF samples and longitudinal assessment over up to 3 years. So we're beginning to develop, to complement some of the other major cohort initiatives globally, a very, very large data set of biological and biomarker data, as well as clinical and cognitive data for analysis. And this is, if you like, the focus data that Paul referred to earlier. I think one of the other key initiatives that's taken place over the last few years are platforms like Dementia's Platform UK, which I'm sure John Gallagher will talk about later, and a new initiative out of Gates Ventures, the ADDI, which Deep UK is also a partner in. This is an environment whereby these data from these large studies will be freely available and accessible for analysis. So I'll just talk a little bit about the EPAD project. This was a huge initiative, 64 million euro grant from the IMI, the Innovative Medicines Initiative, to really try and address what the key issues were in terms of drug development for disease-modifying therapies or therapies for the secondary prevention of Alzheimer's dementia, to really mitigate some of the problems that we've been having in the years. Now, unfortunately, despite the scale of this project, we were unable to actually launch a clinical trial on the platforms I'll describe in a moment. And we can discuss the reasons for that later. But in any regards, what we were able to do was bring together a very large network of partners and clinical trial centers to recruit these incredibly deeply phenotyped research participants. And the objective was, as said, to develop a trials platform for the testing of multiple interventions. There's a platform trial, and we've seen the success of platform trials in terms of COVID therapeutics, concurrently for the secondary prevention of Alzheimer's dementia. One of the key things in this study was the cohort study itself. So what we wanted to do to mitigate screen failure, because again, if I go back to what I said earlier about picking the right population, we've always wanted, I think, to be able to do clinical trials at a much earlier stage of the disease's progression. But those people don't naturally come into clinics. It's very hard to find them in the community. So what we did in EPAD was work from left to right here of building a very large register of 500,000 people across Europe, from whom we selected people we believed to be at higher risk of having Alzheimer's disease, from whom we would then have chosen people who were amyloid positive and other criteria for coming into the proof of concept trial. And this is where, if you like, the 64 million euros was spent in developing this environment across Europe and actually, running the cohort study. Now, one of the challenges I think that we had from the outset of the EPAD project was this issue down at the bottom of this slide, that the decisions that a pharmaceutical company would make about the ongoing development of their asset or their drug was going to be based on a cognitive outcome measure. So we had this debate and this discussion ongoing throughout the course of the EPAD program, and indeed before it, that was it appropriate? Was it necessary? We spoke to regulatory authorities and they said, well, yes, it is. If you want to have a drug treatment for Alzheimer's disease, stroke, dementia, then the primary outcome has to be a cognitive one. So what it meant was that we had to sort of downplay biological manipulation, if you like, or perturbation by a therapy, to see that as a kind of intermediary phenotype on the course to the development of a cognitive outcome. The problem being, of course, that these cognitive outcomes in a preclinical population were at a very great distance to, the effect one might have on amyloidosis or the aggregation of tau. And our sort of Bayesian modeling suggested that, on the R bands, which was our primary outcome measure, in a preclinical population, it would take at least 3 or 4 years before one saw an effect on the R bands, with all the challenges of that tool and the noise in terms of the relationship between, biological change and cognitive outcome. And this was, of course, quite a challenge. And one of the things I think that has really held us back is this reliance on using cognitive outcomes as the primary outcome for clinical trials. So if we continue to perceive neurodegenerative diseases as cognitive disorders, as memory disorders, and I know that not everybody agrees with my statements on this, but I sometimes feel it's like describing cancers as nausea disorders or pain disorders, rather than the actual diseases themselves, which are the target for intervention. It means if we continue to consider Alzheimer's disease as a cognitive disorder, then we'll only really diagnose it when there are overt symptoms coming to the observation of the patient, their families, and then to the clinician. And I think, as I said earlier, that cognition as a primary outcome is acceptable if one thinks about symptomatic agents, but I'm not sure that it's acceptable for a target, for a drug, which is specifically trying to target disease modifications. We're also often told about surrogacy, whereby if a biomarker change doesn't have a relationship with quality of life or well-being or cognition, then it's in some ways irrelevant. And I do argue sometimes, I think that's looking at the problem through the wrong end of the telescope. It should be possible to I think, advocate for the fact that if you modify the course of disease, and you know that disease to be, driving consequential cascades and upstream cognitive developments, then that in itself should be the primary outcome. So just to sort of spend the last 5 minutes or so, just sort of looking at some of the data that we have. So Alzheimer's disease, if we make the assumption from neuropathological studies and from other observations that Alzheimer's disease starts in an area of the brain called the hippocampus, then what one would have to do in this preclinical stage of disease is, in effect, test the hippocampus. And we know from imaging studies, and I'm not an imager, and I'm defer to presenters later on in the course of the day, but we can, of course, with 3T and indeed 7T scanning, look at hippocampal subfields with more accuracy than we were maybe able to do a few years ago. And these hippocampal subfields, of course, subserve, or these areas of the brain subserve particular neuropsychological functions. And we've obviously been discovering over the last few years in animal studies and preclinical studies what hippocampal place cells do in terms of visuospatial memory. So the question is, can we actually test these hippocampal subfields with a kind of an experimental and neuropsychological paradigm? And this is a 4 mountains test. Now, if I were to challenge you with some of the cognitive outcome measures like the R bands or the MMSE, everybody in the audience would do very well to know, exactly where the what the data is, where they are right now, etc. You'd score very well on these cognitive tests, remembering a list, for instance. But this test itself, what I'm showing you here is 4 mountains in a particular orientation. And what I'm going to show you in a second is 4 other images, one of which is this same 4 mountains, but from a different perspective. And you'll be spending a little bit of time looking at these thinking, gosh, this is tough. This is much harder than remembering the date or learning 3 words and recalling them after a few seconds. Now, put you out of your misery, the right answer is A. But this is actually considered to be a very good test of, if you like, these hippocampal subfields for hippocampal place cells. This is a test of allocentric visuospatial memory. Now, from the PREVENT study, which I described earlier, my namesake friend and colleague Karen Ritchie looked at these data that we collected on the 4 mountains test and looked at something called the CADE score, which is it's a good indicator of risk of future dementia, and actually found quite a good correlation between the individual's risk of developing dementia and their ability to perform the 4 mountains test. The mean age in this sample was 52 years old. So already you're beginning to see people at high risk of dementia developing maybe 20 or 30 years down the line, and already evidence of a phenotype which seems related quite specifically to hippocampal function. Now, the issue with this particular analysis was that there wasn't any correlation between hippocampal volume and the Four Mountains test or the CAGE score. However, and I know John's speaking later today, so I hope I'm not going to steal any thunder from him. His group in Cambridge, who have been leading the imaging work on the PREVENT program, were able to demonstrate that there is a reasonably strong correlation between the molecular layer of the hippocampus and ApoE status. So ApoE4 is a well-known risk factor for Alzheimer's disease, Alzheimer's dementia. And what I'm illustrating in this is that if this molecular layer, which is known to be from pathological studies, an early area for the accumulation of neurofibrillary tangles, tau, if you like, maybe we're beginning to see this kind of pathology of tauopathy, which targets a molecular layer in a group of individuals, like I said before, who are in their early 50s, that may be an incredibly good signature for Alzheimer's disease developing 30 or 40 years before the onset of dementia syndrome. And if I orientate you to the bottom right-hand corner of this slide, you know, the ATN criteria, which are finding their way into certainly research practice, that's amyloid, the degree of amyloidosis, whether or not it's tauopathy, neurodegeneration, and then C I've added here for cognitive, is if there's been an assumption it's not measurable in a high-risk preclinical population in early 50s using traditional MRI and cognitive tests. But what I think this work in the PREVENT cohort is beginning to illustrate is that these are now measurable in a high-risk preclinical population in their early 50s using subfield MRI and correlated cognitive tests. And the argument being that if we're unable to measure, for instance, dramatic changes in amyloid and CSF or tau in CSF or in PET scanning, we may be able to specifically measure changes in the molecular layer of the hippocampus correlated with this cognitive function in people in their 50s. Now, I'm not making this as a proposal, but I'm maybe throwing it out as a challenge. Could these become the primary outcome measures for clinical trials of anti-amyloid and anti-tau strategies in people in their 50s, 20 or 30 years before dementia develops? So, in summary, some of the cognitive tests that we've been using, in fact, most of the cognitive tests that we've been using and functional measures have very poor psychometric properties. And I think it's a real challenge to see how a disease-modifying therapy working at the synaptic level of A betas, you know, toxicity to a synapse can be measured accurately through learning a list of 10 words. And I think what we have to do is recognize there's more accurate measurements possible for the biological processes as opposed to the psychological and clinical outcomes. And I think therefore we have to pivot, inverted commas or in parentheses, back to where Alzheimer's had probably hoped we would have been, which is to the neurodegenerative diseases as brain diseases. And this will encourage better science to be applied for disease models, mechanisms of action. James talked about precision medicine and early detection of disease and therefore intervention. An acceptance, I think, that changing disease course is a primary outcome and not necessarily just an intermediary phenotype. So my final slide is, as I think James and Paul were clearly articulating, the value of interdisciplinary work is crucial. In terms of measurement, of course, there's the physics and mathematics of structural and functional imaging. There's the imaging of the cellular and intercellular processes, as well as obviously all the measurement and detail of measurement required in looking at wet lab biomarkers. But I would argue that from what we're hearing already today and probably possibly later today, is that one shouldn't consider this is just for discovery science, but with greater focus at a kind of clinical level and a trial level of disease before dementia, these measurements will be relevant outcomes for clinical trials and practice, and it will help hopefully bridge that translational gap. I think the key problem is that we have to be stronger advocates to the regulatory authorities and that this should be the primary outcome for our studies moving forward. So I'll stop there. Thank you very much for your attention.
Thank you, Craig. That was terrific. And I think you really brought us back from the basic science to how the basic science has impact in the real world. Critically important. So now let's move on to the next speaker before we have the Q&A panel. It's a special pleasure to introduce my very long-time colleague and friend Heidi Johansen-Berg from Oxford, who will talk about imaging in neurodegeneration, opportunities and challenges. Heidi.
Thank you very much, Paul, for the intro, and to the organisers for the invitation to speak. I'm going to focus specifically on imaging, and thanks very much to Craig for his talk is a really nice precursor to some of the points that I'm going to make here, specifically in relation to how we might deploy advanced neuroimaging and neurophysiology techniques in the context of trials and clinical practice within neurodegeneration. So as you will have gathered from James and Craig's talk, and Paul's, there's many different methods and types of neuroimaging and neurophysiological data that we might consider in research and clinical practice. When we think of brain MRI data, there's multiple different types of measurements that we can derive from our MRI scanners that can tell us about brain structure, brain function, brain chemistry, and we've seen examples of all of those. There are other types of imaging methodologies available to us, such as PET, as well as neurophysiological techniques like EEG and MEG that we were hearing about from James, which give us very temporally precise measurements of brain activity with millisecond precision. So what this slide illustrates is that we have a range of different methods available to us. Each of those methods gives us a whole variety of different quantitative metrics reflecting different aspects of brain anatomy, brain chemistry, brain function. And I think most people would accept that these methods have been incredibly powerful in a research setting. So they have generated really fundamental discoveries about the workings of the human brain that have really advanced neuroscience research. But what about when it comes to clinical translation? I think most people would agree that there has been a mismatch between the contribution that these methods have made in an academic research setting versus the contributions that they've made as yet in a clinical setting, in a truly translational setting, and that could lead to the accusation that these are just pretty pictures, you know, all very well for the academics to play around with, but what difference have they made to the patient with dementia, for example? So I'm going to focus on the opportunities and challenges of using these advanced methods in the context of neurodegeneration. So first of all, just want to sort of summarize the key opportunities that I see for use of these methods in the context of neurodegeneration. And one is that they can provide us with really useful insight into the disease processes. And we saw some beautiful examples of this from James's talk, where by using these advanced methods, particularly in combination with biophysical models, we can gain novel insights into the underlying disease process, which then that mechanistic insight might allow the design of novel interventions targeting that aberrant process, for example. Another key way in which these methods might be useful is exactly what Craig was just talking about, to use these as intermediate outcome measures in clinical trials. So as Craig was explaining, the sort of gold standard for many clinical trials is to use a clinical outcome measure. And that's what you need to demonstrate efficacy with in order to take a novel treatment forward. That is the model which is very intractable, that you need to show efficacy of your compound, for example, on the key so-called gold standard clinical outcome measure. And the problem with this, particularly in diseases such as dementia, is that those so-called gold standard outcomes are very unsatisfactory. They're potentially very noisy, subjective, badly behaved measures that, or they're measures that take many, many years to evolve over time. So if these commonly accepted primary outcomes are noisy, are inaccurate, and are slow, that means it's incredibly difficult to prove efficacy of a novel compound. You need enormous trials running over long periods of time. Many of these imaging biomarkers that we will discuss might be closer to the underlying disease processes. If these imaging measures are closer to the underlying pathology and disease processes, potentially they provide us with a more accurate, more sensitive, and earlier marker of efficacy. And if we can shift thinking such that those intermediate outcomes are accepted as primary outcomes in trials, then it means that those trials can be quicker and can be smaller, and progress can be made more quickly. And then the third way in which these types of imaging measures can be potentially useful is for prediction and stratification. So at the level of individual patients, now thinking more in a day-to-day management of individual patients, could we use brain scans, brain measurements to make decisions and predictions about individual patients? So for example, in the context of dementia, could we use an imaging measure to predict whether someone was likely to respond to a particular treatment? Could we use these measures to stratify patients for trials so that we target interventions at the subgroups that are most likely to benefit from those? Or could we use them at the level of an individual patient to predict what the future progression of the disease is likely to be like for that individual? So for example, in the context of dementia, large numbers of individuals present with mild cognitive impairments, with difficulties with their memories and cognition, for example, but only a subset of those individuals will go on to be diagnosed with Alzheimer's disease or dementia. And currently it's hard for doctors to predict of those patients coming into the memory clinics, which are the ones that are going to go on and develop full-blown dementia. And just to illustrate the types of predictions that might be useful in this context, I'm going to show some work from my colleague Gwenelle Douaud, who took a multimodal imaging data set along with CSF measurements to test whether by pooling this information together, you could accurately predict at the level of an individual whether a patient attending to a memory clinic diagnosed with mild cognitive impairment, what's the likelihood that in 2 years' time, that individual is going to be diagnosed with Alzheimer's disease. And what she found was that by grouping these measurements together in predictive models, you got higher accuracy at those predictions compared to just using one of those measurements by themselves. So this is illustrated here for the combination of the brain diffusion measures and structural measures. So 2 different metrics that we can derive from our brain MRI scans. By putting that information together, we can classify or separate the individuals who are going to convert to Alzheimer's disease versus those who won't convert. Having characterized those subgroups within our initial training data set, that means that when faced with a new patient, let's say an individual whose data point appears here where the green triangle is, you can predict with some degree of accuracy which of those 2 subgroups that patient is likely to fall into. So this illustrates the principle, and obviously the types of cohorts that Craig was just talking about, things like the EPAD cohort, will provide invaluable large-scale data over time to make these types of predictive models with much higher accuracy than a single small study can do, such that we can begin to understand what combinations of measurements in a baseline setting can most accurately predict the future course of disease. And a key point is that we need to be able to quantify the success of those predictions at the level of an individual patient, a new individual patient. So ideally, though, we would make those predictions even earlier. So this particular example is based on individuals who've been diagnosed with mild cognitive impairment. Can we push back to make those predictions earlier? You were just hearing from Craig about high-risk populations. So individuals who carry the E4 allele for the ApoE genotype are at higher risk, we know, of going on to develop dementia later in life. And my colleagues, Nico Filippini and Claire Mackay and Paul, some years ago demonstrated that young asymptomatic carriers of this gene already show differences in their brains, in this case in functional activity. So using functional MRI to look at brain activity either at rest on the right or in the context of a memory task, they were able to show this increased functional activity in the hippocampus, that key region for memory function that we know to be vulnerable in dementia. Already this brain area is behaving in a different way in this high-risk population compared to their counterparts. Now in this snapshot, we don't know, the sort of million-dollar question is which within that high-risk population, only a subset of them will go on to develop dementia. We really want to be able to define which of them are going to go on to develop those outcomes. To do that type of, to determine that type of prediction, what we need is large numbers and long timescales. And one potential great game changer in terms of large numbers is the UK Biobank project. which Paul, as well as Steve Smith and Carla Miller in Oxford and many others across the country have been involved in adding brain imaging metrics to UK Biobank. So as some of you will know, UK Biobank was originally a prospective epidemiological study in 500,000 late middle-aged older adults. And a few years ago, an imaging extension to UK Biobank was funded, and which is in the process of bringing back 100,000 of those individuals for brain, body, and heart MRI. And my colleagues derived a very powerful protocol of multimodal brain MRI measures, including many of these more advanced techniques such as functional imaging and multimodal structural imaging and quantitative imaging, which can be acquired very rapidly in about 34 minutes in these 100,000 individuals. And then the key thing about this, this cohort is that these individuals are being followed up over time such that their future health outcomes will be made available. So the objective then is to take this rich multimodal brain imaging data set in combination with information about these individuals' environment and lifestyle and genetics over time, adding in information about their long-term health outcomes, such that then with artificial intelligence and machine learning approaches, you could learn what combination of these features can predict long-term health outcomes. And it really is the scale of this study that makes it unique. So hopefully this video is playing for you as well. You're seeing the scale of UK Biobank, so at 100,000 individuals. It's by far the largest brain imaging study in the world. And the reason that that's really important in this context is that within that group of 100,000 individuals, fairly large numbers will go on to develop disease in relatively quick periods of time. So over the first 5 years or so, about 2,000 of those people would be expected on the basis of demographics to be diagnosed with Alzheimer's. That means you could go back to those brain scans that were acquired before the disease was observed to ask, are there features in those scans that would have allowed us to predict who was going to go on and receive the dementia diagnosis? So it's only by having those numbers that you can get those sort of very, very early pre-symptomatic predictors going outside of high-risk populations. So I think that those types of data sets, which are being made openly available to the research community, will provide a really invaluable resource for modelers and statisticians and epidemiologists to mine those resources in order to identify what are those key predictive measures that allow us to predict future disease. And then the fourth type of opportunity that I want to discuss is how do we move beyond this research context to really using these measures in everyday clinical practice. And this is really, I think, where as yet as a community, we've not done enough to translate the measures that we use every day in a research setting. How do we build up the evidence base and then make that translational jump to use those metrics in everyday clinical practice? Why has that been so hard, particularly in the case of measures where there is now a strong evidence base that a particular brain feature might give us useful clinical information? Why then is that not being used in day-to-day practice? Well, that's partly because of various barriers to translation. And one of them is almost you can almost think of as a cultural barrier. And that's that brain scans are viewed very differently and used very differently in day-to-day clinical practice versus research. So if we take a standard, routine structural brain MRI scan, that's considered in very different ways by a neuroradiologist versus by a research scientist, and that's sort of caricatured here. So the neuroradiologist will use his or her decades of experience of viewing brain scans and understanding the patients and histories. He or she will use that history of experience and their own eyes and brains to interrogate that brain scan as a picture, to look for subtle features and signs in that scan that allows them to make a clinical decision about the likely diagnosis of that patient. By complete stark contrast, the way in which that same scan is used in a research setting is completely different. In a research setting, we treat that scan as a three-dimensional grid full of numbers. We treat it as a quantitative collection of data, which is then fed into quantitative image analysis pipelines and often pooled together with many other types of data to reach some type of statistical decision about how it might vary, for example, relative to some normative population. So the way in which these same scans are being used is dramatically different between these two settings. So if there is added value in using these scans in a quantitative way, what are the barriers to making that jump? Well, a few barriers are here. So one is that to use the scans in this way, in the way that we do in a research setting, one challenge is that often takes time. So it's not a question of somebody just looking at the scan and making a conclusion. It's a question of running all sorts of different processing pipelines, which can be slow and computationally demanding. Another big challenge is that as soon as you begin to use these scans in a quantitative way, you need to worry about standardization. So you need to worry about what the numbers in the image actually mean, and you need to standardize in those in some way such that we can pool scans across multiple different patients, multiple different hospitals, for example. And the challenge there is that different scanner manufacturers, different protocols, different quality of scans, all of these different variables in terms of how the data are acquired might give rise to different numbers in those scans. So we need methods to try to solve that problem of standardization and harmonization. We also need ways to actually extract those quantitative metrics from the data in some way that can then be fed into analysis pipelines. And this is an area where my colleague Steve Smith and others in Oxford have made some advance driven by the UK Biobank and other of these large big data neuroimaging projects, where they've developed routine image analysis pipelines that result in so-called imaging-derived phenotypes, so numbers that you can extract from these brain scans that reflect something meaningful in the data. And a few examples are illustrated here. We also saw examples in Craig's talk of things like the subfields within the hippocampus. So these are all ways in which we can process these brain scans, result in a number which tells us something about a particular feature of interest. So these so-called IDPs, imaging-derived phenotypes, or numbers that quantify some feature in the scan, are now made routinely available in these big databases such as the UK Biobank. And those are the numbers that are made available to the research community so that, for example, epidemiologists can understand what environmental features influence hippocampal volumes. Potentially, these are the types of numbers that could be generated on the fly in individual patients if we wanted to make the jump to using these scans in a quantitative way in a clinical setting. Then what you need would be some way of comparing the subject in the scanner's hippocampal volume, for example, to relevant population norms. And again, this is a non-trivial thing to do because of all of these challenges around standardization and harmonization. How do we know whether a number we get out of an individual patient is normal or not? And again, my colleagues in Oxford, Masood Hussain and Lisa Nobis and others, have done some very nice work with the UK Biobank data sort of towards tackling this problem by coming up with population norms, growth curves, if you like. These are, you can think of these a bit like the baby growth curves that you'll be familiar with if you've had babies, where you have population curves so that you can see where your baby's weight sits relative to the population norms. So these are population norms over the age range for the left and right hippocampus, where you could imagine that if we had the right databases of potential population data with these metrics, then we could, on the fly, generate appropriate population norms for the patient in the scanner using, for example, demographic information, using information about the kind of scan that we're acquiring, generate those norms such that we can then compare the patient in the scanner to what we think of as normal for patients of that type. And in order to push this type of agenda forward, what we really need is closer integration between the clinical services where scans are routinely being acquired and the research community, which again has been a very difficult thing to achieve for all sorts of different practical problems. But my colleague in Oxford, Claire Mackay, has made some real progress in this regard where with the support of the NIHR Oxford Health Biomedical Research Centre, she's been able to establish the Brain Health Clinic. So this is an integrated clinical and research service for memory clinic patients in Oxfordshire, such that when patients are referred for their routine clinical brain scan, those scans are being delivered by one of our research scanners, where the protocols have been optimized to be able to both provide the standard clinical information that the neuroradiologists are used to wanting, but also have been optimized to be able to be harmonized to the UK Biobank data, such that the types of population norms that I was just showing you would be able to be generated, so that we can develop reporting tools so that we can extract quantitative metrics from the scans being acquired in the individual patient and compare those to population norms in order to provide quantitative reports to help inform the clinical teams. And in order to create these types of integrated clinical research services, one needs to consider all sorts of practical issues around things like governance and ethics, but processes can be set up such that, for example, patients are given the opportunity to consent for their routine clinical data to be used for research purposes, and also the opportunity to consent for additional measurements to be taken, such that we can use this type of facility to test, to generate an evidence base on new innovative imaging metrics. And this, we think, is a sort of win-win all round, in that on the clinical side, the clinicians receive higher quality imaging data with the potential for follow-up and improved access to studies and trials. On the research side, it provides us with a translational interface for novel measurements, for example, as well as participants with good run-in data for studies and trials. And also from the patient's perspective, they're given a more thorough assessment than they might otherwise have got, which potentially could give rise to an earlier and more accurate diagnosis, and are also provided with more opportunities to participate in research. So in summary then, advanced neuroimaging has delivered valuable insights into discovery in neuroscience and mechanistic research in a disease context, but true clinical translation is a work in progress. And areas of potential translation include insights into disease processes that can guide treatment development, intermediate outcome measures in clinical trials, patient stratification and individualization of therapy, and prediction of future disease. But realizing this potential depends on the development of robust, accurate, and clinically feasible methods for quantification, as well as integration between clinical and research field. And this is very much an area that requires the bringing together of the physical scientists who will build the models and the quantification tools with clinicians and neuroscientists who can interpret what those measures might be reflecting. So finally, I'd like to thank colleagues and funders, and thanks very much for your attention.
Heidi, that was terrific. Thank you very much. May I just ask all of the other panelists to come and be prepared to join me on the virtual panel here this morning. We've had lots of really interesting questions, and I'm going to dive right into it to make sure that we use our time well. I think the first one I want to start out with touches on this key question of developing, whether we should focus on, well, if we focus on intermediate phenotypes, how we can use this in practical ways most powerfully. I think there were a number of kinds of questions that touched on this, and one of them was, what are the challenges in using intermediate phenotypes in medicine if we find measures deep of hippocampal substructure, measures of amyloid and others? Why are regulators, what is the rational basis for regulators' caution in embracing intermediate phenotypes? And then the flip side of that is, let's just pose it as a question. What kinds of things do we need to do conceptually to link intermediate phenotypes to patient benefit? Is it always through our standard cognitive testing, or could we think more broadly about what interdisciplinary science, modern physics, and sensors could deliver? So could I ask maybe Craig, if you start with that, and then just a quick, some additional comments from James and Heidi.
Yeah. Well, I think it's and Heidi used the term the gold standard, and the gold standard at the moment, of course, is cognitive behavioral functional measures in neurodegenerative disease. Those are the patients we see. Those are the concerns that they have. And I think I was trying to argue that intermediary phenotypes should be endpoints in themselves, not intermediary for something else. And I draw often on other branches of medicine, where, for instance, let's take cardiovascular medicine, for instance, an amount of, coronary artery atheroma would in itself be a target prior to the individual having a heart attack. And a good outcome for that patient would be a reduction in occlusion of the coronary arteries. And I think in neuroscience, we need to start thinking about the disease of the brain in the same way. I mean, if I'm a 50-year-old with amyloidosis, however we discover that or detect that through clinical practice, would I be happy if somebody was able to remove all that amyloid from my brain? Now, I think I would be. So James is shaking his head, but I think if we have a belief that abnormalities in tau, abnormalities in amyloidosis are relevant for later development of a brain failure, then should that not be the target in and of itself? And I think that's more of a sort of philosophical, clinical discussion.
So let me just challenge you, and then James can pick up on this point. So I guess the question Martin Rosser was posing, I think, to summarize it, is whether, let us say, amyloid, a hallmark, is necessary and sufficient to mark those people who go on to develop clinical disease.
Just very quickly on that point, I think a lot of the analysis we've done, and I saw, Martin, your comment in the Q&A, has been cross-sectional. And the cross-sectional analysis has been done in older people with dementia. What we need to look is, and we will do this in studies like PREVENT and EPAD and others, is look at longitudinal changes of those key biomarkers or those key pathologies and be able to correlate them with maybe some earlier clinical symptoms, visuospatial memory, mood, sleep, other probably relevant phenotypes earlier in the course of disease. Quite interestingly, we published a paper with my PhD students, Lucy Sterling, looking at the EPAD data, and actually found the higher the level of comorbidities, the less likely you were to be amyloid positive. So there's a lot of stuff going on in older people with comorbidities and amyloid, which I'm not sure we fully understand. And I do wonder if amyloidosis in very late life is not as relevant in terms of Alzheimer's disease than it is earlier in the course. But I think we need to, that's something we need to explore.
James, what can you add to the thoughts?
Yes, so just to pick up on Craig's phrase, I think we need to get away from this statement of belief that it's simply enough to treat amyloid at early biomarkers and actually really have concrete evidence, because there's a history to this, to a mistaken adoption of biomarkers. At the early days of MS trials, it was assumed that reducing the number of acute plaques would reduce disability with treatments. That turned out not really to be the case, and I think led to an unfortunate digression in many trials. Or the use of statins and their impact on cardiac and cerebrovascular disease is largely independent, or substantially independent, of cholesterol levels. So they are effective, but not for the reasons that people initially believed. So it's really important that we develop proper validated causal models that link early causes to outcomes that matter to patients. And we don't rely on statements of faith. If we, for example, if we don't understand how and why, even whether those early pathogenic processes actually lead to outcomes that matter, then simply treating that molecular genetic cause, if I put it that way, cannot be assumed to have an impact on disease in a way that matters to patients or health economics. So we really do need to have much more rigorous causal models.
I just think that I agree with everything you said. I just think the problem we face is almost one of time course. that if we are going to be able to intervene against what may or may not be, but let's assume with belief that they are relevant disease processes, we would want to do it 20, 30 years before dementia develops. By definition, these people are not patients.
That I'd agree with. As we do for stroke and heart prevention, we treat blood pressure and cholesterol in one's 40s and 50s. That's normal thinking now. Doesn't seem to be such a problem. In other branches of medicine, this is well-trodden turf. The regulators, I think, are quite supportive of this. I mean, I touched in the written discussion, that this is a really urgent problem. So, for example, in the genetic frontotemporal dementias, there are several compounds now that have really good evidence of their plausibility to what I might call neutralize the effect of those genetic mutations, to either halt or even prevent the genetic FTDs. But since we don't know how the gene leads to dementia, motor neuron disease, Parkinson's disease, or no symptoms at all, we cannot use clinical outcome measures at the moment as outcome measures, because we don't know what the outcome would be. And that's over and above, I think your problem is if you take somebody who's healthy at 50 and treat for 5 years and they're still healthy, how do you know if you've done any good? I mean, that's sort of how you touched on it.
So let me move us on. I think that you both highlighted is some of the inevitable tensions between in use of intermediate phenotypes in the absence of full mechanistic understanding of disease. Now, Heidi, let me come to you, because you've obviously thought about holistic data integration models, where one is looking at very broad sets of data from potentially from the body as well as the brain. We've had a number of questions about about the challenges that have come to our to a previous neurocentric model of dementia. And Craig, I couldn't help but notice something that I'd never seen in the Alzheimer's drawings before, and that is that there are lipid vacuoles in cells that he illustrated. We've actually forgotten about those as being part of what he saw as a hallmark. So, that gets us back to atheroma and atherosclerosis, and cardiovascular disease. Heidi, can you talk about, how models can help us develop quite holistic views, integrating many different modalities, and what we need from our physics friends to do this better?
Yeah, I think that's a very good point. And resources like UK Biobank are really making it easy for people to see the value of those types of holistic approaches, because they're making these large multimodal, multi-organ measures available to the research community. And I think it's increasingly clear across the whole of medicine that these sort of multimorbidities and these effects are often incredibly important, whereas our conventional ways of studying diseases tend to be incredibly focused. I think another driver of this approach to help change the way we think about things in the context of brain disease will be many of the efforts now to try to understand COVID and long COVID, where clearly this is something that is that we don't yet understand, that the community is working together with, and that lots of different multimodal, multi-organ measurements are rapidly being collected. So I think one good outcome of the current situation we find ourselves in is that is forcing those different communities to work together to try to understand something from scratch. And perhaps that will, both in terms of the culture, but also literally in terms of tools for pooling together very different types of measurements into these integrative models, might help to then provide an approach that could be, which could then be rolled out into other diseases, which we currently are treating in a more neurocentric way, for example.
So that really highlights how, in some respects, the field is shifting, as Craig was pointing out, that we're starting to look to large populations of currently healthy people to understand the genesis of much later disease, and in doing so, we can look broadly. We can look at issues like gut-brain interactions, cardiovascular brain interactions, and so on. Now, this challenge of that James has highlighted regarding intermediate phenotypes, or what is the true causal model of a complex disease like Alzheimer's or frontotemporal dementia, has been, I think Fred Wilson is highlighting this with his question: he's asking if, I'm afraid it comes down to a little bit of detail, have any of you seen a paper in recent Science Translational Medicine from Sanchez, which I haven't, that Fred describes as showing a time course of tauopathy progressing into beta deposition, amyloid beta deposition, rather than reverse. I don't know, James, do you have any comments on that? Did you see that?
I've seen that discussed at a meeting, but I've not seen the Science Translational Medicine paper yet. I'll have to dash off and see that now it's out. I think that's really interesting about the complexity, the temporal dynamics of these multiple processes and their interactions. Of course, those interactions may change over the decades. The way forward, I suspect, is to get away from arguing about this process versus that, my biomarker versus your biomarker, and for the field to move jointly to a more mature cooperative framework. We've seen that, I think, in the UK is especially well placed for this in its dementia landscape. The highly successful model of COVID working in collaboration has just illustrated that the power of this. And it'll take us, I think, to more interdisciplinary methods. I mean, particularly with relevant to the diversity of the audience today. Once we move away from classic clinical outcome measures, it's not just about the role of a patient-facing neurologist or psychiatrist in clinic. It steps back and it brings in those who can bring new statistical approaches, new physics-based tools. We've seen some of those in the talk this morning, as well as that clinical edge. So it becomes a much richer and interesting, hopefully more productive endeavor.
No, it's a really interesting issue. So I've just quickly looked at this paper. Fred, thank you for pointing this out. I think it highlights that there still is much to learn. It does remind me that there's lovely work from Bart de Stroper that has shown that it's in amyloid expressing animal models. It's actually human glial cells that are needed or human glial cells independently can induce tau expression in neurons. So that really highlights that the relationship between amyloid and tau is not a simple one, and it's not a neurocentric one, but is a cellular system driven approach. And it is possible that some of this question and the answer to the question lies in unraveling this complexity as it varies over brain regions. Let's just go on. We have a couple of more minutes only, and I want to raise another issue that seems really relevant to this interdisciplinary forum. Engineers and physicists have developed a remarkable array of tools to sense lots of things in our daily life. Where we move, how we breathe, how we speak, their analytical approaches for text mining. I mean, I'm just wondering, and perhaps you could all comment on this, whether we can look to a new revolution in describing phenotypes in people we currently think are pre-symptomatic. Is it possible that they're not really pre-symptomatic? It's just that neither they nor anyone else have noticed the symptoms because they're too subtle. And we could use better analytics to find signs and or symptoms. I'm struck, I always remember Iris Murdoch, and who, as you may know, developed dementia. It's been people have done text mining of her writings, and one can begin to trace the development of it in terms of word choice and grammar, literally a decade before the clinical expression. So let me ask you to comment on that and put a challenge out there for the engineers, physicists, and mathematicians who are with us today. Heidi, do you want to start for just a minute?
Sure, yeah. I think it's a hugely exciting area of sort of mining day-to-day digital data, which is such an enormous resource these days. And I think, evidence on those early signs in word use, early signs in gait or mobility, which might precede any types of cognitive signs and which potentially could be detected with very, very, cheap light touch technology, or even just through our usage of everyday technology, mobile phones being the obvious one. So I think that is a phenomenally exciting possibility. obviously raises enormous amounts of sort of societal and ethical questions around how one would gather that data and mine it and use it effectively to identify warning signs in individuals. But I think the evidence base that the way in which our day-to-day, everyday behavior would give early clues through our interaction with the world, you know, where we go, what we do with our phones, how we move, there are early signals in there. We do have the technology to detect them. It's a question of how that could be plugged into doing something, intervening at the level of an individual.
Good. James, do you want to comment for just a minute? You're on mute.
So there, sorry, I mean, just to really to echo what Heidi said, I think she's put it extremely clearly that you know, this is, I think, a change in the field we've seen this morning, but you know, I'm very optimistic that this can be made to work, but it will require interdisciplinary effort and collaboration and new ways to both image the brain, but more importantly, to understand the data that we're getting.
And Craig, let me give you 30 seconds for a clinical word to highlight how much we need our friends from other disciplines.
Absolutely. I mean, I think that I'm very excited about speech as a biomarker, and there's a lot of work going on. There's a paper recently that IBM actually published with colleagues in America about some differences in speech patterns between MCI and healthy controls. And we put a lot of work in that area actually on top of PREVENT to look at speech patterns. I think I think that will be a key focus. There is the ethical issue of speaking to Alexa and then her telling you to go to the doctor. I think we have to get our head around that. But I really think in that preclinical population, like I said, it's a long silent period. But I think if we listen better, we'll hear some signals coming out from individuals and intervene and hopefully prevent dementia.
Fantastic. Well, listen, there's still many questions that are outstanding. All the speakers will try to type in some answers over the next little while. Let me thank all of you. It's been a great panel. Steve, would you like to lead us into the break?
Thank you, Paul. And on behalf of all of us, I'd like to thank you and all our speakers this morning. If ever there were, I cannot think of better examples of the value of interdisciplinary approaches to the grand challenges in medicine than the talks this morning. They have been absolutely outstanding. I'm grateful to all of you. And can I say, as a physicist and addressing my own communities, maths, physics, and engineering, there are some cri de coeur there. There's some numerous opportunities, particularly, may I say, around the mathematics of causal models. We need to bring new mathematics to the party, I argue. We need to apply mathematics from rather different fields to this territory. There's numerous opportunities, and we'll be exploring some more of those as we follow. So thanks again to all, and to you, those contributing to the Q&A. We will reconvene at 10 to 12 after a short coffee break with Carl Friston in the chair. See you soon.